Alright, welcome back to more biology revision. Okay, so we have tonight, tomorrow, and then that's it. That's paper one. Honestly, I'm feeling like a bit half and half at the moment. I feel okay about this paper. Paper one is definitely the easier one in terms of overall content, because we've had 2 years to go over it, but still, I was doing a couple past papers, and there really are a lot of mean questions that they can ask. Some really, deceptively worded questions. So, that's what I want to do today. I want to go over the extended response questions. The 5 and, I think, 6 mark questions that we could get asked about specific processes, specific practicals, everything like that. So literally, the way I'm going to do this is I'm going to start off with paper one, and I will literally just go through every single year and go only through the extended response questions. We'll obviously write them out here. So let's start off with 2023. And we want to be looking at the standard print. Okay. Paper 1, June 2023, and we'll pull up the mark scheme. Okay. So I'm just going to flick through, and we're only going to look at the 4 and 5 markers that aren't to do with, that aren't to do with practicals or anything but will rely on graphs or context. The first one here is, describe how a quaternary protein is formed from its monomers. Okay. So, 5 marks. Every time it's a 5-mark question, you want to get at least 6 points. Point 1. It's saying that a quaternary protein is formed from its monomers. The monomers are obviously going to be amino acids. So, it's going to be a condensation reaction forming peptide bonds between amino acids. So already it's saying how it is formed from its monomers. This is how it's formed. It's condensation forming peptide bonds between amino acids. Now we can delve into the more complicated stuff. The primary sequence is determined by amino acid. So the primary structure is determined by amino acid sequence. Secondary structure is determined by hydrogen bonds. Hydrogen bonding. That's how it's determined, because it can form an alpha helix or a beta pleated sheet. Tertiary structure is formed by different interactions in the R group. For example, ionic bonds, disulfide bridges, ionic bonds, disulfide bridges. There's one more thing that I'm forgetting. Hydrogen bonds, just on their own, possibly. And then quaternary is more than one polypeptide bonded together. So, let's see how many marks. Describe how a quaternary protein is formed. From its monomers. We've said monomers, we've said how it's formed. Okay. Amino acids joined by peptide bonds, 1 mark. Amino acids joined by peptide bonds, 1 mark. Gonna get rid of his pen. 2 marks. By condensation reaction. One thing I should have mentioned here, which I didn't, but I know would be worth a mark, in many cases, is if you're talking about condensation, always mention that it is the removal of a water molecule. I lost like 2 marks in one past paper because I literally just didn't mention that one term. Removal of water molecule. Okay, so 2 marks already. 2 marks out of, this is 5. Okay, no mark for talking about the primary structure, but that's fine, so no marks there. Secondary structure is formed by hydrogen bonding. That's exactly what I said. Accept alpha helix or beta pleated sheet for secondary structure. Okay, so 3 marks. Tertiary structure formed by interactions between R groups. For interactions, you could say hydrogen bonds, disulfide bridges, ionic bonds. Okay, cool. And finally, quaternary structure contains more than one polypeptide. That's exactly what I said. Okay, so 5 marks. That is how you would answer that extended response question. It asked us how they are formed. So obviously we mentioned condensation, we mentioned the forming of peptide bonds, we mentioned the monomer. Make sure to do all of that before going on to talk about how it becomes quaternary. Ow. Obviously the only thing we could have done that wasn't in the mark scheme, but it's still important to remember, condensation is always the removal of a water molecule to join 2 monomers together. Okay. Describe the structure of DNA and the structure of a chromosome for 6 marks. Oh shit. Okay, right. Right, we'll start off with DNA. Could we draw a diagram? Will it allow us? Let's have a look. Let's draw the phosphate group. It's meant to be a ribose. Is ribose a hexose or a pentose sugar? I don't remember. Anyways, ribose and the base. It's kind of difficult to draw it in reverse, I can't lie. Obviously, you would do this with a pencil. So that would be your DNA. So DNA, the structure of DNA, is a polymer of nucleotides. And we're talking about DNA here. DNA is a polymer of nucleotides. We will then go on to say what that is. It's nitrogenous base, phosphate group, and our deoxyribose sugar. Then we will obviously say that phosphodiester bonds will join adjacent nucleotides, and that hydrogen bonds join complementary bases or base pairs. So adenine to thymine, thymine to adenine, cytosine to guanine, guanine to cytosine. Okay, so we have talked in depth there about the structure of DNA. The structure of a chromosome is DNA associated with histone proteins. It is tightly coiled to form chromosomes. And I don't remember anything else. The word supercoil rings a bell, but DNA associated with histone, tightly coiled to form... I don't know. I don't remember anything else but we need to know about the structure of chromosomes. So let's take a look. Let's take a look at how many marks this is. It's a polymer of nucleotides. Word for word. Polymer of nucleotides. 1 mark. Nucleotide consists of deoxyribose, phosphate, and an organic / nitrogenous base. Phosphodiester bonds between nucleotides. DNA, double helix, held by hydrogen bonds. Or 2 strands held by hydrogen bonds. Okay, I didn't mention that. Hydrogen bonding between adenine, thymine, and cytosine, guanine. Okay. So we could have actually got 2 marks out of this. The other mark point would have been... Okay, so hold on. 1, 2, 3. 3 marks so far out of 6. We could have pulled 2 marks out of this. Hydrogen bond forms the double helix, which obviously makes sense, but like, that just seemed a bit... I just kind of forgot that was a thing. Hydrogen bonds form a double helix and complementary base pairing. Never in an exam write A, T, C, G. Always write adenine, thymine, and cytosine and guanine. That's what it wants you to say. So, you know what? I will give myself a sympathy mark here, because I did say what it said. Hydrogen bonds between bases. Ignore bases identified with letters. I know that in an exam I wouldn't do that, so I will give myself the benefit of the doubt though. Anyways, DNA is associated with histones / proteins. 5 marks. During mitosis, chromosome consists of 2 chromatids joined at a centromere. Okay, I did not put that. So, out of 6 marks, we got 5. Pretty decent, honestly. Considering a vast majority of that question was about the structure of DNA, I have a feeling a lot of people would easily skip over that, would easily gloss over this. You know, they would say, oh, DNA, they would probably say that, but I feel like not a lot of people would delve into the structure of DNA as much. And very easy mark point to miss about DNA, how it's the double helix is held together by hydrogen bonds. Very easy to miss mark point. Is there another question? Yes. See, these are the kind of questions I need, because if the paper is out of paper's out of 90, paper 1. If paper 1's out of 90, then these extended response questions are going to be worth, what, 1/6 of the paper? If I can guarantee every one of those marks, then that's going to be really helpful. That's basically the same as getting like a level 3 or a level 4 response in the essay question. If you can guarantee that you get all of these questions right, that's going to massively help your grade. Right, this question. Mutation can result in an increase in genetic variation. Describe and explain the other processes that result in increases in genetic variation. Oh, shit. What are the other processes? Oh, independent assortment, crossing over, fertilization of gametes, fusion of gametes is random, I think. 1, 2, 3, 4. Okay. Fusion of gametes is random. Number 2, crossing over. We obviously will have to explain this. The way I remember it, I don't remember it. That's why I'm kind of panicking. Crossing over It's where your homologous chromosomes associate to, it's a bivalent. It is, they form a chiasmata? How does that chiasmata get formed? I'll literally just put it. Chiasmata forms, then it's equal lengths of allele slash, would it be like, I'll just put equal lengths of allele are exchanged. And this occurs during meiosis 1. Then it's independent segregation. This is literally where, during meiosis, what the hell have I just spelled? This is literally where, during meiosis 1, the chromosomes will go to opposite sides. Is it literally just that they will assort randomly, increasing variation of assort randomly, increasing variation in gametes? Right, let's look. Independent segregation of homologous chromosomes and pairs. Really? All you had to say was independent segregation of homologous chromosomes. No details. No details. Wait, so does that mean I just wasted a crossing over between homologous chromosomes and pairs? Oh my god, you didn't need to explain it. All you would have needed to say is between homologous chromosomes. Oh my god, how have I fumbled this question by going too much into detail? That is crazy. Okay. And then random fertilization of gametes. Accept random fusion. Okay. Best case scenario, I got 3 marks out of 4 for this. The one thing I missed is produces new combinations of alleles. Obviously, the question asked, describe and explain the other processes that result in increases in genetic variation. Obviously, that's going to produce new alleles. That's a nice term though. You could really milk that term. Produce new alleles. You get asked a question about genetic variation, just milk that word. It's the same shit that you do when talking about enzymes. How you have to milk the word enzyme substrate complex. Because apparently they just love when you use that word. So, 3 out of 4, 5 out of 5, and 5 out of 6. So we got 13 out of 15. Fairly decent. We only dropped 2 marks. Rarely those 2 marks with tiny details. Very tiny details. The 2 details being hydrogen bonds hold the double helix together, that's easy. One being new alleles increases genetic variation. New combinations of alleles, sorry. So, let's go on to a different year. Let's go on to the 2022 paper. And again, it's likely that I've done these questions before, but let's still do it. Okay, this is a 5-mark question. Describe a transport of carbohydrate in plants. I kind of put... Why have they spelled it like that? The transport of carbohydrates in plants. This is going to be the mass flow hypothesis. So, because my hand hurts, I am literally just going to write like bare notes here. I will round up marks if I think I get any marks. Okay. Firstly, the carbohydrates produced from the source are going to be actively transported out of source via companion cells into the phloem. Because they are insoluble, because they are soluble, this is going to increase, no. Think about the water, decrease the water potential in the phloem. Therefore, water from the xylem is going to move via osmosis, you have to mention osmosis, into the phloem, down the water potential gradient. This is going to increase turgor pressure, so the carbohydrates are going to move down the pressure gradients into the sink. And just very quickly, in case it's worth a mark point, mention where they can be used as cellulose for structure, as storage as starch, or can be hydrolyzed into glucose for respiration. That should be 5 marks. Sucrose active... Okay, you have to mention sucrose. Ouch. Really? Okay, honestly, I would have done that because I have wrote maybe 5 times now an essay about the mass flow hypothesis. Every time I've used the word sucrose, it threw me off it talking about carbohydrates. It really that really threw me off. So I don't know whether that would be a mark. But they are actively transported into phloem via companion cells. 2 marks. Lowers water potential in phloem, and water enters from xylem by osmosis. So that's all 1 mark. Produces hydrostatic pressure. Produces hydrostatic pressure gradient. Really? We get told it's turgor pressure. Accept description of gradients. Okay, well, it says hydrostatic in brackets, so let's just tick it. Increases turgor pressure. So, mass flow to respiring cells. Accept young leaves, fruit, shoot. Unloaded/removed by active transport. Okay, we actually missed a lot of details here. Number 1, obvious thing, always refer to sucrose, not carbohydrate, because that's not a thing. It has to be a polysaccharide. Number 2, increases turgor pressure. You would then say that it is mass flow to. Then you have to say where. I guess some mark schemes you're allowed to say source and sink. I guess you're not in this one. Which kind of sucks balls, but okay. The zinc could be respiring cells, fruit, storage tissue, anything like that. So what I basically said here, just in more detail, that will be how you get all marks. So I got 4 out of 5. I'll write that down. So, again, only way I could have improved that, refer to it as sucrose, elaborate a little bit more about how it ends up in the sink, and also, little detail I missed, it's then actively transported into the sink. I didn't know that. Makes sense, but I didn't know that. So not a bad question, actually, because I would have gotten a lot of that wrong. Compare and contrast the structure of starch and the structure of cellulose for 6 marks. I've just done this with my teacher. I've literally just done this with my teacher. Also, it seems like there's always 3 extended response questions at the back instead of 2. I always thought it was 2. Okay. So, compare and contrast the structure for 6 marks. Number 1, in fact, we'll literally just do it like this, because I've already answered this. You know, I would take my time with this in an exam. That's all I'm going to say. Right, starch and cellulose. Most obvious differences. Starch is a polysaccharide made of alpha glucose. Cellulose is comprised of beta glucose. Now, I don't know how you would word that, whether you would say it's a polysaccharide of. I guess you could say that. So that would be mark point 1. Mark point 2. Cellulose is long/unbranched. Oh, both have glycosidic bonds. But I don't think that's a mark point. I think they have to reject that if you say long and unbranched. Starch has amylopectin, which is branched. Starch is used as a storage molecule, so it's insoluble. Cellulose is used, I guess you could also add both are used in plants. Cellulose is used as structural support for the cell wall. Starch has amylose, which is helical and tightly coiled for compact storage. Cellulose has hydrogen bonds that form cross links, which form microfibrils. That's all the differences I can think of. Let's have a look. Both are polysaccharides. Really? No fucking shit, they're polysaccharide. What the fuck? Really? That's a mark point? Okay. Both contain glycosidic bonds. Yes, 1 mark. Good eye. Both contain carbon, hydrogen, and oxygen. What? What is this mark scheme? What do you mean? Starch has alpha glucose. Okay, 2. Starch is helical and coiled. Cellulose is straight. Yes. 1, 2, 3, 4. Cellulose has microfibers. Starch does not. Okay. So, in an exam, make sure you compare. Because just saying they have microfibers is not enough. You would have to say starch does not. Really though, that's insane. Saying that starch and cellulose both have CH and O. I mean, we should have gotten all 5 marks, but if the examiner is picky, which they are gonna be, that's not very nice. Complete digestion of starch in a mammal. Okay, that's kind of mad. Complete digestion of starch. So it's amylase, which is going to hydrolyze. So amylase enzyme. Hydrolyze starch into sucrose. Oh, I don't know this one. Let's have a look. Hydrolysis of glycosidic bonds. Oh, it's maltose, not sucrose. Okay, that's where I went wrong. Right. Hydrolyze starch into maltose. I would have gotten a mark for saying hydrolyze, but I missed such an obvious mark point. It hydrolyze breaks glycosidic bonds. Also, didn't need to put salviary. Starch to maltose. So, starch to maltose via the action of amylase. Maltose to glucose by maltase. And I think that would have been it. Okay, cool. So I would have gotten that if I remembered that it became maltose. Because I was sat there like, wait, it's not, is there a thing called sucrase? I was like, what the hell? I don't know. It's because I completely forgot. So, I would have got 1 mark for that, let's be real. Better way to format this would be hydrolysis of starch into maltose, breaking glycosidic bonds via action of amylase enzyme. Then you would go on to talk about it becoming maltose, maltase, glucose. That was a fairly challenging question because I forgot. I actually forgot. And that could have been those 4 marks could have been the difference between like a grade A and a grade B. So let's make sure we keep on top. Right. Describe how mRNA is formed by transcription in eukaryotes. Oh, it's so boring. If we get a question about transcription and translation, I'm probably going to get it wrong because I've had to do it so many times that I'm that bored. How mRNA is formed by transcription. Okay. It's going to be RNA polymerase breaks apart hydrogen bonds between complementary bases. This is going to unwind the DNA molecule. Then free-floating RNA nucleotides binds to complementary bases. I'm just kind of, yeah. Adenine, is it adenine? Thymine, adenine will bind to uracil. Binds to uracil. RNA polymerase phosphodiester bonds between adjacent nucleotides. I'm giving up. And then splicing of pre-mRNA to remove introns. Okay. I'm so tired. Hydrogen bonds between DNA bases break. No mark for saying unzipping. Okay, cool. Free RNA nucleotides aligned by complementary base pairings. Yes. Adenine binds to uracil. I should have said RNA uracil binds to adenine on DNA. I would have said that in an exam. Oh yeah, you could have just said uracil used in place of thymine. RNA polymerase joins adjacent nucleotide by phosphodiester bonds. Yes, 2 marks. Pre-mRNA is spliced to remove introns. Yeah, easy. Easy as pie. RNA, so hydrogen bonds are broken apart between complementary base pairs. Free-floating RNA nucleotides will bind to their complementary base pairs. Adenine will bind to, I guess you could literally just say uracil used in place of thymine in the RNA nucleotides. RNA polymerase forms phosphodiester bonds. We get it. We've done this question like 52 times. It's that boring that I'm actually now making mistakes on it because of how many times I've had to write the same question. If the next question's boring, I'm going to give up. Oh, it's so boring. Describe how a polypeptide is formed by translation. Let's look at the mark scheme. mRNA attaches to ribosomes. tRNA anticodons bind to complementary mRNA codons. Yes, I would have put that. tRNA brings specific amino acid. Yes. Then you would say amino acids are joined together via peptide bonds via a condensation reaction using energy from ATP. Yes? Joined by peptide bonds with the use of ATP. Aha! The one bit I wouldn't have got. tRNA released and the ribosome moves along the mRNA. But that's easy. Oh, this is another question. Define gene mutation and explain how gene mutation can have no effect or a positive effect. Okay. Gene mutation is a change in the base sequence of a gene. Correct? Change in the base nucleotide / base sequence. You could have also said results in the formation of a new allele, which I didn't. It has no effect on an individual because DNA is degenerate. It means multiple codons will code for the exact same amino acid. Therefore, no change will occur to the protein. It will have no effect. It could have a positive effect if the mutated protein is advantageous. Therefore, the allele is advantageous, increasing the chances of an organism surviving, reproducing, etc. So it has no effect because genetic code is degenerate. Has a positive effect because it results in a change of a polypeptide that positively affects the protein, may result in increased survival. Okay. For 4 marks, you could easily get that. Just say a mutation results in the formation of a new allele because there is a change in the base nucleotide. Honestly, guys, that's all that I really can... Oh, hello, someone is watching. Oh, someone's been watching for a while. I'm so sorry. I thought it was DNA helicase. DNA helicase is for DNA replication. We've always been taught it as RNA polymerase. I can quickly check it if you like, but I'm... Yeah. Yeah, it is. So, that's the thing, because we do a lot of shit in, like, the paper 2 topic about, RNA polymerase and its role of actually, like, starting transcription. Man, I do not have any more time, do I? Okay, well, I just googled it and it's given me the most piss-take answer ever. RNA polymerase does not break hydrogen bonds between the sugar-phosphate backbone. No shit! What? What kind of AI-generated slop is that? What? Instead, it breaks hydrogen bonds between the complementary base pairs. Yeah, okay. What kind of AI slop answer is that, by the way? Does not break hydrogen bonds between the sugar-phosphate backbone. Yeah, because it's phosphodiester bonds. That's funny. Very quickly, just because, like, I'm bored and I have nothing better to do with my life, I will go to the 2017 paper. No, apparently I won't. It won't let me. I want to look. I want to look. I want to look at more extended response questions, but don't bore the living shit out of me. Past papers. But yeah, it's not DNA helicase. DNA helicase is for semi-conservative DNA replication. It is a like common thing to get misread though. Like, you know, it's a very similar process, except obviously with transcription. And one thing I forgot to mention actually when I was writing that, like, you know, one thing I forgot to mention in semi-conservative replication, both strands of DNA are gonna act as what's the word, template strands. In transcription, only one acts as a template strand. Right, AQA paper 1. Right, we'll look at the 2018 paper, and then that's me going, because I'm too tired for this. And I'm a lazy fuck. Okay. I'll literally just scroll to the bottom. We'll see what questions they are. If they're nice, then we might answer them. Okay. This is 2018 though, so this might be... Okay, yeah, this is like ancient shit. Describe the gross structure of the human gas exchange system and how we breathe in and out. I'm guessing they will give you like half and half here. You give a couple of points about inhalation, exhalation, and the gross structure would just be talking about like the trachea branching off into... Oh shit, I don't even remember. The bronchi, the bronchioles, and then the alveoli? Describe a gross structure. What does it imply by that? Is it wanting you to go into specific detail, or is it wanting you to just name the components? I guess it's time to look. Name structures. Tracheae, bronchi, bronchioles, alveoli. Above structures named in correct order, or in a diagram. Okay, interesting. So 2 marks. Would have literally just been worth 2 marks to say trachea, bronchi, bronchioles, alveoli. Yes, okay. 2 marks. Breathing in, that is when your diaphragm contracts, diaphragm muscles, and it's always your external intercostal muscles contract. That will increase the volume in the thoracic cavity. reducing pressure, therefore establishing a pressure gradient. Atmospheric air will move in. Breathing out is just the opposite. The diaphragm muscles will relax. Internal intercostal muscles will contract. Or you could literally just say external intercostal muscles relax. Causes volume to decrease. Okay. Honestly, I would have read that question and panicked, but it wasn't that bad. 2 marks for literally just naming the trachea, the bronchi, the bronchioles, the alveoli. 2 marks for that is crazy. And then 4 marks for literally just talking about gas exchange. That's easy. Please be a nice question. Mucus produced by epithelial cells in the human gas exchange system contain triglycerides and phospholipids. Compare and contrast the structure and properties. Oh, shit. That's kind of a tough question. Not tough as in difficult. Just like, that's a cool question. You could probably get a diagram about this. We'll draw a diagram. If I can remember what one looks like. Literally just saying that, I mean, look, it's like spot the difference. There's an organic phosphate here, there's not one here. They both have glycerol. They both have fatty acid chains, except this one has an unsaturated and a saturated. This one is all saturated. This can associate to form a bilayer. I don't know about this one. I'm going to go out on a limb and say no. Obviously talk about how this is the polar region. That's the non-polar. How this is hydrophilic. That's hydrophobic. And I don't know much else about triglycerides, to be honest. Oh, obvious one. Both contain ester bonds. That would have been such an easy mark to get. I feel like in an exam at this point, I would know to put that, because the amount of times I've lost marks for not saying the bond is crazy. I've lost so many marks in so many past papers for not mentioning the word ester. It's crazy. If you're being asked a question on how a phospholipid is formed, obviously mention condensation, and then always mention the bond. So, both contain ester bonds. Fatty acids on both may be saturated. Oh, really? I always thought these were always saturated, and that was un. Huh. I didn't know that, genuinely didn't. Both are insoluble in water. Again, that wouldn't be one that I put, but that makes sense. Both contain CH and O. Come on. That's such a piss take of an answer. It was the same with the cellulose question. Like, no shit, they contain carbon, hydrogen, and oxygen. Oh, come on. Triglyceride has 3 fatty acids. Phospholipid has 2 plus phosphate. Yeah, I would have got that. Triglycerides are hydrophobic / non-polar. Phospholipids have a Oh! I didn't know this. I didn't know that triglycerides are entirely hydrophobic. And this is different regions. Okay. Honestly then, that was kind of a fun question because I didn't know half of that. I know about the fatty acids, how there's 2 there, 3 there, phosphate there. Wouldn't have got many other mark points. Out of 5 marks, I might have gotten 3 marks. Describe how lactose is formed and where in the cell it would be attached to a polypeptide. Oh, okay, wait. This is a nice question. I like how they kind of give you a bit of a story with these questions. It's like, oh, mucus. How lactose is formed. So that is obviously going to be glucose plus galactose. You should say condensation. Obviously elaborate on what that is. Removal of a water molecule. Forming. I don't know why I put the G like that. Forming, shit! Why can't I remember the name of the bond? Glycosidic. Then it's in the Golgi apparatus, where they are checked and modified. Where proteins are checked and modified. So let's have a look. Glucose and galactose, joined by condensation, joined by glycosidic bond, added to polypeptide and Golgi. 4 marks. Okay. Not bad. That was not a bad question at all. See, I kind of like how the 4 mark questions are always really, like, easy. I've noticed that. Every time there's 2 big extended response questions, the last question's always really easy. Like, the mark scheme is always really lenient, which is kind of nice. The other 2 are a bit of a nightmare. I probably would have failed that question about phospholipids because I didn't know half of those things, but now I do, which is good. That question on breathing, that would probably also be one that I did okay on. So, yeah, I'm happy to wrap things up there. I'm very happy to wrap things up there for today. I am tired, and hopefully the extended response questions we get in the paper in a couple of days will be fine. Because, let's be completely real, I feel like it won't be bad, like the extended response questions. Most of the time, it's either telling you to compare something. It's either telling you to describe a biological molecule, or it's telling you to describe a process. That's what I've noticed. Every time it's a 5 or 6 mark question, it wants you to go into detail about a process, or it wants you to explain a biological molecule. So remember the structure of DNA, remember the structure of cellulose, remember the structure of glucose and shit like that, because you could lose marks just for forgetting basic things, which I've done. I mean, having to literally state the elements made, like, is comprised of glucose is crazy. But anyways, it should be fine. It should be fine. I will obviously be live tomorrow. Probably not for as long, because I obviously want to, like, save a bit of time, but what we'll do tomorrow is we'll go over some extended response questions. There's a couple more papers left. And then I want to go over, like, the 1 and 2 markers. The questions that are, like, really odd. Where, like, you know, they'll ask you something that you learned, like, a year ago and you completely forgot it. That's what I want to go over. For example, there was a question on 2023 paper. Why are viruses considered non-living and acellular? And it's like, what does that mean? Like, obviously, acellular is, you know, as the name implies, but having to explain that is kind of odd when you've never answered it before. So I would like to go over questions like that, just to make sure I don't forget any little details. And then, yeah, that should be us done.